Hello and welcome. My name is Funwie Nchobnjong, pharmacy student at Chicago State University. Today, I will be doing a presentation on Treponema pallidum, causative agent of syphilis for microbiology and immunology. I will narrow down my discussion about Treponema pallidum on the following subheadings: physiology and structure, pathogenesis and immunity, epidemiology, clinical disease and syndromes, laboratory diagnosis, treatment, prevention, and control. Now let's start with the physiology and structure of Treponema pallidum. Treponema is a gram-negative spirochete. The bacterium measures about 0.1 by 0.2 by 6 up to about 20 micrometers in length. Treponema is thin, tightly coiled, and has pointed straight ends. It has 3 periplasmic flagella inserted at each end. Treponema does not replicate in vitro due to the absence of Krebs cycle. As a result of no Krebs cycle, the bacterium relies on its host for purines, pyrimidines, amino acids, and other essential nutrients. This is an electron micrograph of Treponema pallidum. Looking keenly at the photograph, you will notice the spirochete nature of the bacterium. Another interesting thing about Treponema is that it is microaerophilic, or it is almost anaerobic. Thus, it is extremely sensitive to oxygen toxicity. Its genome sequence revealed Treponema has no catalase or superoxide. These enzymes are the enzymes that are responsible for converting toxic forms of oxygen to less toxic forms, and this explains why the bacterium is very sensitive to oxygen toxicity. Clinical disease and syndromes. As I earlier noted, Treponema is a causative agent of syphilis. The clinical cause of syphilis evolved through 3 stages or phases, which are the primary, the secondary, and the tertiary phases. Primary syphilis. During this stage, an initial syphilic canker develops at the site of penetration of the spirochete. Syphilis lesions may appear within 10 to 90 days. A characteristic canker may develop within 1 to 2 weeks. The ulcer may heal within 2 months, giving the patient a false sense of healing. Secondary syphilis. Secondary syphilis is usually characterized by flu-like symptoms such as a sore throat. There may be headache, there may be fever, there may be myalgia, anorexia, lymphadenopathy, that is swelling of the lymph nodes. There may be generalized macular, papular, or pustular rashes, which may emerge but may again heal in a month's time. Highly infectious raised lesions called condylomata lata alongside mouth erosions then appear. I would like to reiterate again here that these raised lesions are very highly infectious. These symptoms may disappear within a few weeks and the patient enters a latent phase. Tertiary syphilis. Tertiary syphilis is also known as latent or late syphilis. Usually, patients progress to this phase if they go untreated. The tertiary syphilis is characterized by an asymptomatic period of years up to about a decade. In this asymptomatic period, patients usually suffer multiple organ damage leading to blindness, dementia, arthritis, and this is because the spirochete penetrates these organs. For instance, it may penetrate the eye, it may penetrate certain parts of the brain, explaining blindness and dementia caused by the bacterium. Granulomatous lesions called gummas may be found on the skin, bone, and other tissues. Congenital syphilis can lead to serious fetal diseases such as latent infections, multiple organ damage. There could also be malfunctions in neonates. Pathogenesis and immunity of Treponema pallidum. Once more, Treponema pallidum is the causative agent of syphilis. The bacterium has three subspecies, which are the Endemicium subspecies, which causes endemic syphilis in certain parts of the world, the Petennius subspecies, which causes yaw, which is a skin and bone infection, the Caratium subspecies, which causes pinta, another skin infection. It is difficult to study its virulence since it cannot be cultured in vitro in high concentrations. Usually to study the virulence of Treponema, high concentrations of the bacterium is required. And since Treponema cannot be cultured outside living cells, it is therefore difficult to obtain high concentrations in order to study its virulence. However, genome analysis revealed that lipoproteins anchored in the bacterial cytoplasmic membrane are not exposed to the outer membrane, making it possible for Treponema to avoid the immune system. If you recall from our study of immunology, the immune system is only able to mount an immune response when it recognizes certain proteins as non-self. However, in the case of Treponema, since the bacterium does not expose lipoproteins on its outer membrane, the immune system does not recognize it as non-self in order to mount an immune response against Treponema. And this is how Treponema is able to avoid the immune system, causing different disease states in susceptible hosts. Treponema can resist phagocytosis and instead adhere to host fibronectic tissue. This adherence permits interaction with host tissue, causing damage. Currently, there are three speculations about the tissue damage as a result of Treponema. It is speculated that tissue damage as a result of Treponema could be due to hemolysis, higher urinodasis, or as a result of the host's own immune system trying to fight the infection. Histology of syphilis lesions reveal infiltration by polymorphonucleocytes, leukocytes, or macrophages. Epidemiology. Syphilis is a global bacterial infection. It is the third most commonly transmitted bacterial infection in the United States. Fortunately, the incidence of syphilis decreased in the 1940s as a result of the discovery of penicillin. Unfortunately, in recent years, there has been an increase in the incidence of syphilis up to about 50%, and this is typically in the male homosexual population. Treponema pallidum can be transmitted by direct sexual contact with an infected partner, via infected IV transfusions, or congenitally from an infected mother to a fetus. This is a chart from the Centers of Disease Control and Prevention showing the incidence of syphilis as a result of different sexual behaviors in the United States. Looking keenly at the chart, you'll notice that the dark blue is the male homosexual Caucasian population, which is the highest risk group. It is followed by the African American male homosexual population, and lastly by the Hispanic homosexual male population. Laboratory diagnosis. How can we diagnose Treponema in the lab? There are two different techniques, serology and dark field microscopy. Dark field or direct fluorescent antibody staining microscopy could be used to identify the spirochet. Serology test is either specific or non-specific. Non-specific or non-treponemal test measures the immunoglobulin G and immunoglobulin M antibodies against lipids. The most common are Venereal Disease Research Laboratory VDRL and the Rapid Plasma Reagent RPR test. The specific treponemal test dictates specific treponema pallidum antibodies. Examples are fluorescent treponemal antibody absorption FTA-ABS test, the treponemal pallidum particle agglutination TPPA test, and the enzyme immunoassay EIA test. Treatment, control, and prevention. How can we treat, how can we control, how can we prevent Treponema pallidum so that we can be free from syphilis? Currently, penicillin remains the drug of choice against Treponema pallidum. Long-acting benzathine penicillin is used for early stages of the disease. Penicillin G is recommended for late stages of the disease and as well as for congenital syphilis. Doxycycline and tetracycline are alternative therapies for patients with penicillin allergies. However, for those patients with neurosyphilis, penicillin must be used for treatment. In such cases, the patient must be desensitized against penicillin allergies so that they could be adequately treated. At the moment, there is no vaccine available against syphilis. Therefore, safe sexual practices and adequate treatment of infected sexual partners remain the best control measure for Treponema pallidum. Syphilis cannot be transmitted by inanimate objects such as toilet seats or household utensils. These are some few references. The Centers of Disease Control and Prevention remains the best option for further information regarding Treponema pallidum and syphilis. Thank you so much. I hope the information conveyed in this presentation has given you some insights about Treponema pallidum and syphilis, as well as how to control, prevent, and treat syphilis.